Welcome to the Proteomics in Proximity podcast, where your co-hosts from Olink Proteomics talk about the intersection of proteomics with genomics for drug target discovery, the application of proteomics to reveal disease biomarkers, and current trends in using proteomics to unlock biological mechanisms. Here we have your hosts.
Hey there, welcome to Proteomics in Proximity. I'm your host, Cindy Lawley, with my co-host,
Dale Yuzuki.
Today we're talking to Karsten Suhre from Weill Cornell Medicine. He's calling in from Doha, Qatar, where he's been driving collaborations for over 10 years. He's here. He's going to talk to us about integrating genomics data with various intermediate phenotypes like metabolomics and proteomics. He has recent publications on, he's got so many publications, it's ridiculous. He does meta-analyses on COVID, obesity, translation of biomarkers to the clinic. And if we go far enough back, he's got some publications on atmospheric science, which I am just fascinated by, coming from an oceanography background. So this man who exists in boundary conditions between sciences and really overcomes barriers for analyzing these data is so exciting to bring on board. So, Karsten, with that introduction, would you like to just start off by telling us what you'd like our audience to know about you going into our discussion?
Yeah, all right. Thank you very much, Cindy and Dave, for having me. I mean, you already covered so much about what has been going on, so I would just say let's just go into it right away. And
okay,
I understand.
Questions.
Go ahead,
Karsten,
Dale.
I understand that you came from a background in physics, like atmospheric physics, theoretical quantum physics. What caused you to go into the genomics field?
Well, I mean, it was a chance event. I initially did quantum field theory, went to England, learned about fluid dynamics, like that very much, tried to do a PhD there, but then I moved basically into atmospheric chemistry. And then at some point, I went back to industry for private reasons and had to go back to working with engineers and stuff like that. And then at some point, I thought it's much more interesting to be a researcher rather than an engineer. I wasn't fitting into engineering. And at that point, I just by a chance event, I read the Le Monde where they talked about the human genome. And I just had the chance that the CNRS actually allowed me to change field when I returned to France and just
CNRS is
CNRS, Centre National de la Recherche Scientifique. That's what do you want to say? The National Research Center in France, and they do basically everything. And I was
this was maybe 2001 when the draft was announced. That kind of thing was what you saw.
Exactly. And I was supposed to go back to atmospheric chemistry, but I thought, well, this is interesting. And then I came across a lab in Marseille where basically they were all physicists, astronomers. Bioinformatics at the moment didn't exist.
Yes.
And bioinformaticians were basically physicists.
So he was learning the language. He was learning French. He already knows German. He knows English clearly, and he's learning the language of biology. I'm saying it's not that easy.
Well, also back in 2001, right? The human genome had just been finished in a draft form. This is when GeneBank was still in a library of 15 CDs, compact discs, right? And this was at a time when people's comprehension of genes was growing, but we had no idea how many genes there were, right? Estimates were in the 100,000 genes, et cetera, et cetera, et cetera. But to have a draft and all of a sudden, okay, now what, right? What was that like?
Well, at the time we worked on 1,000 genes. We did bacterial genomes. At the time you could publish one genome in Science.
A single genome, yeah. Right. And as far as that at that time goes, what did you find about that transition from, atmospheric chemistry, help me, is that like ozone layer type of work or is that other kind?
Also, yeah. I mean, we actually have a Nature paper on ozone in the upper atmosphere.
Of course you do.
On aircraft. It doesn't even show up in PubMed because it's not medical.
That's funny.
But in the end, it's this. After that, actually, I didn't really change fields. The thing is, if you go for atmospheric chemistry, what we did is we measured all kind of chemicals in the atmosphere using aircraft and using ships and balloons and lidars and things like that. And then when you integrated the data, and that was my job, basically integrating all these measurements, I like to say it's the same thing as what we do before. The only difference is the organism is the Earth. You have atmospheric chemistry.
It's epidemiology,
like
right? Dealing with big data, messy data, figuring out what are your cutoffs, what are your outliers, and what to believe.
And the systems biology. I mean, the kind of modeling of the data. We had differential equation systems that are the same that are used today to model metabolomics in cells. It was just the metabolomics in the atmosphere, and things were transported around by
This
fluid
is
dynamics.
great. The systems biology of the Earth versus the systems biology of the human. Yeah?
Yeah, and you would be surprised that there's so much match from one to the other. You could match all the chemical reactions, the stuff like that. There's so many problems that are exactly the same.
It's the atmosphere of the human.
Of course, it's a different dimension.
The atmosphere of the human. I love it. Yeah, it makes total sense.
Yep. And therefore, then at CNRS, then you got involved in the genomics piece. Can you tell me what it was like in 2002 to 2005 in those early years? I mean, that was HapMap, right? Cindy and I were at...
No, not even HapMap. I mean, when I started, it was a lab in Marseille, and they were working on bacteria genomes. So they were some of the first of sequencing them and actually integrating them with structural biology. So whenever there was a gene discovered, the team crystallized the protein, overexpressed it, of course, did the structure, and then tried to understand the function. So it was functional bioinformatics. The human stuff only came in 2006 when I moved from Marseille to Munich. Then suddenly I moved from bacteria to humans with the Cora study.
Yeah, that Cora study. What an amazing study.
What is,
Dale, you and I were in Illumina by then. You and I were...
Help me with the Cora study. I'm not familiar with that.
Yeah. Well, the Cora study is a population study. So basically it's, you know, maybe the Framingham study. So, and Cora is the German Framingham study, if you want. Like thousands of people recruited, every phenotype, blood drawn, re-recruiting, things like that. And the Cora study at the time was just the right moment because all this omics started off. So they were genotyping them. They were starting the GWAS in 2006. The first GWAS came out. And then where we were is like we were the first to actually run a GWAS with metabolomics at the time with Biocritis and Metabolon, the two companies. And from there on, actually, if you go further on, came the idea to do GWAS on all these kind of intermediate phenotypes, the different metabolites, proteins, you have it. And that was really very, very interesting thing. And everything I did since then is basically a scaling up of what we did.
And that particular association, right? The GWAS studies that we're familiar with are GWAS to disease. You're bringing in a new layer, and you're calling it an intermediate phenotype. Can you explain that a little bit more?
Yes.
What is an intermediate phenotype?
Yeah, I mean, okay, I should pay credit to the person who really brought up the idea. That's Florian Kronenberg. He was also formerly Munich, then Austria. So the idea of the intermediate phenotype is to say you have the genome that actually produces your effect, your protein levels, your metabolic level, and then that affects the disease. So if you want to know how a genetic variance goes and determines a disease endpoint, you can say, okay, this gene has a risk for diabetes or whatever, but you don't know really why and what's the function of the gene. The moment you put a phenotype in between that is basically an outcome of your genetic variation and at the same time a cause for the downstream effect on the disease, you're much closer to your phenotype and you have a much stronger statistical signal once you do your association studies. And then, of course, you can build up networks out of that, right? You can go from transcriptomics to proteomics to metabolomics. You could throw in protein glycosylation, phosphorylation, everything you can measure on an omics level. And then in the end, link it to the endpoint. And I think nowadays, the concept of Mendelian randomization very much gets into that as well. To say you have this intermediate phenotype is basically like what people think as exposures. And then you have the the outcome, which is the disease.
I want to just say that, when Karsten and I spoke when I was doing metabolomics, him telling me that this story was very impactful, just like blew my mind, that an intermediate phenotype in power, like I hadn't thought about the power of it, but it's a bit of a magnifying glass to be able to help you see, by improving power, help you see what's really going on in the connection between disease and genetics. So I just wanted to honor that explanation, because I think you do a great job of translating information into a biologist's perspective.
Well, in the end, it's all statistics, right? And when you use the word power, it's really well defined, and it's the way to say, if I do a study in a human population, I have a lot of noise. I mean, there's a lot of, it's not monogenic mice, so getting the signal out of the noise is what is your statistical power. So what chances do you have to get a statistically significant signal? And an association with a complex endpoint like diabetes needs much larger numbers than an association with a metabolic intermediate phenotype. So you can go stepwise from the gene to the metabolite, and then associating the metabolite with the endpoint. And as I said before, nowadays you can put in many randomization and even ask questions like, is it causal? Is it worth being targeted by a drug, or is it rather a biomarker?
In 2005, right, I'm selling the first human microarrays, whole genome microarrays, to the National Institutes here in Bethesda.
Yes.
And then after the Selects acquisition, started selling genome analyzers for looking at all this variation.
And don't skip over the work we did with the NHBLI in order to do the Framingham cohort, which was certainly one of, I remember staying up till 4 a.m. writing that grant, Dale.
Well, I was going to say that, all that genomic work of whatever, I mean, from 2007 for the first genome analyzer coming out, to the present, right? So much genomic data, right? First thousands, then hundreds of thousands, now millions of genomes and exomes, and yet so little impact upon such common disease like you mentioned diabetes, right? Where there isn't a genetic signal. But what you're saying now is genetic variation with the intermediate phenotype, we can understand how people get diabetes. Is that correct?
Yes, that's the point. And also you can basically dissect the pathways. So if you have now 100 different genetic signals on diabetes, they don't all associate with in the same way with the same metabolite. Some may go through more a brain pathway, an endocrine pathway, whatever. And the metabolites that are associated with these variants, they can tell you which pathway things are going and what's actually happening. So in the end, I also there's an image, it actually comes from Biocrates. They had this first on their slides to say, in the end, this intermediate phenotype is like an imaging. So they had this kind of image of a dog that was in very broad pixel. And the deeper you go into there,
more resolution,
the more you see. So in the end, I think intermediate phenotype is like an imaging of your biochemistry and what happens in your body. And that's why it's so worthy to go for this. And
therefore, you're getting very unusual connections to diabetes then, like you mentioned, in terms of the systems biology piece.
And you have an opportunity, I think, also with a non-Northern European population to characterize biomarkers that will then be moved to the clinic in order to monitor diabetes where triglycerides or HbA1c aren't successful, right? That's the exciting aspect to me, is being able to identify new ways to monitor or to predict those that are at a higher risk for developing diabetes years in advance.
But now you're talking about the Qatar biobank, right? When you say non-European populations and things like that. I mean, that's a very, very important point as well. I think something that's nowadays also linked to this intermediate phenotypes. And I think there, if we come back to, for instance, what Olink is doing in UK Biobank, is this quest, especially of industry, to say we want to find human knockouts like PCSK9. So we would like to find a human homozygote who is hopefully living well, and then try to figure out why is that, what does this knockout do, and is it something that can be targeted, that can be beneficial, and things like that. And we just did an analysis on the Qatar Biobank, and it turns out because we have this kind of very consanguineous population, basically you have 160 times chance of finding a human knockout in the Qatar population compared to UK Biobank. And that, of course, makes it even more interesting to go for people where you have like a rare homozygote associated with extreme protein levels, low or high, or extreme or low or high metabolite levels and things like that. And then from there on, it's not GWAS anymore, right? It's basically like these individual cases. It's not even rare variant. It's really like you want to have the homozygotes that are there and see, are they on PCSK9? Is this very low LDL? Is
and that sounds like a very low-cost study. Once you've been able to identify those knockouts, you can do it in a small number of samples, but you have to have the genetic data. Is that fair to say, or does it take a lot of samples to test that? I just am thinking about GWAS, right? GWAS, there was so much we couldn't see in those first GWAS studies back in 2005, 2006 that were done with the GWAS arrays.
I mean, you still need big numbers to find these.
In the genetic data, sure. But once you've got those genetic data, I guess my question is structure of the study when you're layering on proteomics could be a smaller study because you've already done the genetic, you've identified the heterogeneity and characterized your extreme phenotypes, or what you expect to be your extreme phenotypes.
You're right. You could do that, but I would probably still go for the whole cohort on terms of proteomics and metabolomics, because in the end you don't know who you want to. You don't want to start with the genetics and then
messaging.
Just do a subset.
I love that.
Karsten, would you mind backing up a bit and tell me a little bit of higher level view of this particular study or this particular cohort in Qatar that you're working with?
Well, I mean, I just said Cora is the German Framingham,
and I do want to say Annette Peters is the current PI of the Cora cohort there out of Augsburg, right? She's in Helmholtz. Annette Peters. I just want to give her a shout out. But back to the Qatar Biobank. Yeah.
So how large is it, and how long has it been collecting samples?
I think when I came here, they were building it up in collaboration with Imperial College, and actually there were also colleagues from Munich who were coming over here as consultants, so I got in contact with them pretty easy. I think at the moment they are well above 20,000. I know that they have 20,000 individuals fully genome sequenced. We ran 3,000 samples on an in-house Metabolon platform here in Qatar, also 3,000 on a SomaLogic platform. And we started working on that end. I think there's a huge potential there to potentially do the same thing as what is happening in UK Biobank, finding maybe someone who's ready to put in funding to get more omics data and things like that. So it's pretty exciting. And as I said, this specific genetic specificity, of course, makes it very valuable to discover these human knockouts.
To be clear, then, it's agnostic for disease. In terms of you're looking at 20,000 individuals, the metabolome, the proteome.
Like Framingham, it's just a cross-sectional thing. Of course, Qatar has a very high diabetes rate, which is a challenge for Qatar. That's why Cornell is so engaged here in Qatar. But it's also, of course, a chance if you want to study diabetes, because you have a very high, there's over 20% diabetes prevalence in Qatar. And you have a lot of undiagnosed diabetics in there, which is also scientifically very interesting, because these are people who are not treated with medication. So if you do all the omics on them, the problem if you want to study diabetes with omics is especially in Cora, sometimes the people with diabetes, they're much more healthy than the others, because they know they have diabetes, they look at their cholesterol, and they do everything they need. And then it's very hard to actually study anything bad about them.
A motivation component, because they've got this sort of, this deadline by which they need to reverse whatever was going on before. That's really interesting.
Yeah. No, I remember there were leaflets from diabetes associations in Germany. They actually said, don't worry, diabetes is not necessarily something negative. I mean, of course, it's a disease, but if you take it the right way, it could actually change your life in the right way.
Prolong your health span, right? That's fascinating.
Yeah, I mean, I wouldn't go and say I want to get diabetes. I mean,
but it does speak to the power. It speaks to the power of feedback, right? And some of these companies that are trying to provide a way for you to monitor your health that's not going to be paid for by insurance, at least not in the U.S. But it speaks to if you identify something really early that looks like you're nudging, you're out of normal or healthy range, then there's a motivation factor to nudge it back, right? It's a wearables argument, maybe.
Yeah, and especially in this diabetes field, of course, the problem is if you're actually able to change things, because there are some things not everybody can change like this, and there's a lot of brain component in this as well. It's not just like, oh, you're fat, you lose weight. Yeah. I mean, it's much more complex than that.
And it's interesting, the diabetes, heart attack or heart health connection, because I was just reading a paper last week that talked about a particular drug on market for diabetes that reduced the risk of heart failure. And it's just remarkable, right? In terms of, we get back to systems biology. We get back to, we have a connected systems. Yeah. And as far as then this particular study goes in that you're working with the metabolome and the proteome, is it you're now looking for funding, or you said that it's like the UK Biobank and that it could lead to something like that, or?
Well, I mean, it's not me.
Yeah,
Qatar Biobank is not a Cornell thing. Qatar Biobank is from Qatar Foundation. So actually, I try to motivate people to say, do this kind of a deal. It would be a win-win for everyone. But it wouldn't be up to me to decide. I see. That.
And moving on a little bit, I think I'm aware of this paper that was recently published in Frontiers in Immunology. What can you tell me about it? This is identification of robust protein associations with COVID-19 disease based on 5 clinical studies.
Yeah, I mean, that's a bit an outcome of, the COVID has changed a lot of stuff also here in Qatar, and it made us collaborate strongly with New York. Because, especially Weill Cornell, New York, they are affiliated with the New Presbyterian Hospital, and they were totally on the front line at the time. And they collected samples from COVID. We collected samples here in Qatar. And my colleague Frank Schmidt, who's running the Proteomics core here, and he has an Olink platform at the moment, he brought basically in these samples, and we measured for Cornell in Qatar and New York these samples. And then asked this question I think everybody was asking at the time, what is special about COVID? What can we learn by doing things? And there was a big rush, and I think there's also a lot of not so good papers out there, so you have to really be careful of what's there. And I'm not saying ours is one of the best. It's just like to combine the data. And at the time, I think Olink, and you can maybe help me on that, they shared of their 1500 platform, a large data set.
Yep, publicly available, downloadable, an MGH study with several collaborators.
Yeah, exactly. So I see 306 cases, 78 controls from Massachusetts General on the Olink Explorer 1536. So it was a very broad study. And then the other 4 studies that you mentioned from Imperial College of London and, like you mentioned, New York Presbyterian, maybe 50 to 200 cases or so. But nonetheless, quite an interesting data set. Yes, Cindy, you're going to say something?
Oh,
yeah. Mm-hmm. Yeah, and that was a bit the motivation to say for us it was an opportunity to really learn about the new Olink platform with the 1500, bring the data together, and the idea was to say, okay, especially with COVID, the conditions are so uncontrolled because the situation was just like, oh, grab every sample you have there, but you cannot make sure that everybody is under the same conditions. To say, if you combine 5 different studies, to say what comes out and was really in all studies at the same time, which doesn't mean that the others are not relevant hits, but these are the ones that are really so robust that even under worst conditions, you can see them. And that makes them very interesting of course, as markers
pathways
and to guide further research. Yeah.
Because we had some that were plasma, some were plasma EDTA, some were inactivated with heat, others were inactivated with Triton X, a detergent, some weren't inactivated at all. And I'm like, so you combined all these different centers, different storage conditions. I don't have information on how they were stored or how quickly they're at room temperature or what have you. What can you comment on that? I mean, from the reviewer's point of view, right? Don't they raise eyebrows when you're like, what are you trying to do here? There's too many variables involved.
Yeah, right. And I mean, it's the same thing in many papers, that there's always concern about study limitations and things like that. But I think as long as you acknowledge these limitations and you say, and you go for the really strong associations, and of course you must make sure that you don't create bias by having confounders. That's the risk. You have to make sure that there's not confounding. But once you're sure about all that, then having something that sticks out of this noise is really a strong signal, and it's really worth following up, because you're sure it will be replicated in the next study, and not it will disappear because the next study is maybe not clean as the first one.
And the abstract of this paper indicates 13 proteins, right, were significantly associated with COVID infection, right, compared to controls, and that these differential expression of these 13 proteins was across all 5, which I think is pretty, like you mentioned, it's a strong signal.
And it was Bonferroni corrected, so corrected for multiple testing as well. So it's not like 0.05
it's much lower.
And stuff like that.
-8.
Why don't you comment on that important point, which is the Bonferroni correction and how many signals disappear?
And that frustration. Yeah, but it needs to be done, right?
Well, it needs to be done. I think the point with p-values is always, if you have a really good study that has enough, and the word power comes back, then you can allow yourself to really go for the strongest hits and focus on them first. If you don't have that, then you have to do p-value gymnastics, things like that. Although you should be aware, of course, there's a lot of more signal below that you don't want to lose, right? Later on, once you know that your study is good and that what are the caveats, you can look at the hits that are not that strong and see, are they biologically relevant? Can I take them maybe as a starting point, as preliminary data for the next grant, and then reinforce that and confirm it? So I'm not saying you should only look at Bonferroni significance, but coming from the GWAS field, it's even worse. You have to be Bonferroni and replicate, which a statistician would say, it's evident it should replicate. But even then, it not always does replicate. And that means it's always good to be very conservative on these p-values if you don't want to end up with things that that just don't work and don't replicate. Because that's frustrating if then you follow up on something and then things don't replicate and you just turn data up and down and just refuses to replicate.
Mm-hmm. One of the interesting things about the results, right, in terms of these 13 proteins, maybe some familiar suspects like IL-6 and interferon gamma. What can you comment in terms of then the practical usefulness of these inflammation-related proteins and its association with COVID?
No, you put me on the spot, because that's really a medical thing. I of course, I try to figure out a bit what's interesting about these. You may also be a bit more reductiveness and say, well, maybe there's a lot of stuff you would not even be surprised about to find them. Is it? And I think that's an important thing. We have other papers where we compare not COVID case control, but COVID against bacterial RRDS. And you ask yourself, what is really specific to COVID? Because just to say there's a cytokine storm, and that's something that could also happen if you have a bacterial infection. I think what really changed something, about COVID in the last years is what we learned about how to treat COVID. And I'm not a specialist in interpreting this. And I find it sometimes interesting what people actually see in our papers, which I didn't even see. And it would help doctors, I think, to understand what kind of access you really want to go in your treatment. I think my understanding is that a lot of why the death rate in COVID went down over the months and the years, in part is because people understood better how to actually treat it. And this kind of information, I hope, contributed to this kind of understanding, at least to a certain level.
That's really helpful, because to be clear for this audience, ARDS is acute respiratory distress syndrome, right? And this is where, right, you mentioned bacterial infection will bring about this,
Yeah.
But COVID will also bring about this, and there's a lot of...
Yeah.
Go ahead.
Oh, I was just...
It's basically like a control, because you always want a control, but a non-COVID person is not a good control for a COVID person if you want to know what is specific to COVID. So then a person who has the same symptoms but not from COVID, that would be a much better control.
And that's the MGH, like the Philbin, Michael Philbin's study as well, right? These were patients that came into the hospital with symptoms that seemed COVID-like but tested negative. Their controls were just that, which I thought was... worked well with what you were doing.
Now, as far as then the proteomic analysis goes, with these particular signals, is there a particular translational message in terms of applying this knowledge clinically? Or is it still too early, right? When people have severe COVID, it's already known, it's diagnosed, they're monitoring for this. Like you mentioned, it suggests new treatment ideas, new clinical trials. I mean, is it...
I was just going to add on to that question. And who might pick this up from those that you work with? Who might pick this up and take it over that line, right? It's, is it Gabby? Is it others who just not to put you on the clinical spot,
No. I think the point is, in this case, we would probably not even know, because the people who really pick it up would be the doctors who are capable of reading these papers and translating that into practice. And maybe, I don't know whether it's maybe too dangerous, but maybe let me comment carefully on this. You may have heard about Didier Raoult.
Can you...
He's the person.
Give some background.
He's a French doctor who promoted the use of proteome in
now I remember.
COVID.
No, I'm not. Yeah.
Yes, actually. So no, I'm saying treading carefully. I don't want to make any statements here. I've been working directly with him. We worked together when I was in France, and we sequenced bacterial and stuff like that. I think the way when he said this works in my hands. That he was criticized a bit against whether his studies really worked or not. But knowing him, I think he was a bit like, he treated patients in a way that he knew how to work with these patients. I think the knowledge he as a doctor personally had, and that may come out of which chemokines and cytokines and whatever work, I think in a way he may have treated patients the right way and treated them. What didn't work out was this statistical thing like black and white, proquinone or not. And that's a whole different story. But I think my feeling he was very honest on that end, that he probably for himself treated this patient and saw an effect on that. And now coming to this translation thing, I think it's actually based on this kind of knowledge. So I think interacting with some doctors is like, there's a lot of doctors don't really work like a robot. Like, say, if there's A, then do B, and then do C. There's a lot of more like, what's the whole feature of this patient? And that's where the omics comes in, and where I think where the bridge is still not there. How do you really bridge that in a way? You cannot necessarily nowadays easily bridge it. Maybe artificial intelligence at some point, algorithms, things like that could do it algorithmically. But I think there's a lot of stomach knowledge from doctors, where they learn from these studies, they take something from there, they have their own picture on what's going on, and then they treat patients on that basis. And it's very hard to quantify and translate that into a case control research paper, and that's where maybe Didier Raoult get into some trouble, because people expected that to be a case control study, and you have.
Is it the role of the MD PhDs perhaps to do that, or translational scientist is a job description I see much more commonly now, right? And I'm trying to imagine what are their objectives, and what are their key results that they're evaluated on, and I hope that it's helping facilitate this translation, because I think that when a scientist throws it over the fence, it may not it may not land in a place where a person can carry it then to the clinic.
I think there's a huge gap between what we do on the research side and really getting it to the doctors, because the doctors in the end, they want to have something that's actionable.
Replicated,
and just telling them here are 50 proteins that go up and down is not something they can really translate. And that's really the big thing where I think we've been working a lot on, and I hope things are getting better. But if you want to go to personalized medicine, I mean, you cannot go at the same time to double-blinded clinical trials, but it's this N equals 1 thing. If you want to treat one person, you cannot do a test on 100 before you decide on the one.
And the doctors have to make those decisions, which is always seemed challenging to me.
Yeah, one of the interesting points you bring up is the art of medicine versus the science of medicine.
Yeah.
And like you mentioned, in terms of treating the individual patient, true personalized medicine oftentimes go with gut decisions based upon decades of experience in treating patients with similar symptoms. And then it's, well, okay, what is that art? What is it about this patient?
Yeah,
How do you capture that?
Yeah.
Exactly.
Yeah, and I think that's where of course all this omics stuff, if doctors learn about that, if they can integrate that into their gut feeling in a certain way. And then of course this gut feeling, if you can, and then with the other buzzword you can, if this gut feeling could be translated through artificial intelligence or whatever kind of things into something there, that because in the end you don't want to have like 2 doctors in the world who can cure everybody and the others don't know what to do, right? You want to basically have every doctor be able to treat every patient.
And going back a bit to a topic we touched on a little bit before in terms of that intermediate phenotype multi-omics kind of approach, it seems like your work now primarily focuses on proteomics and metabolomics. You mentioned, well, you can go into glycomics, right? You could go into transcriptomics.
Methylation or epigenetics.
Methylation, I'm curious, particularly on the transcriptomics piece, your perspective now in terms of the usefulness of RNA-seq, either at single cell or bulk level, as it intersects with this intermediate phenotype idea, because obviously, right, DNA to RNA to protein, is it very useful signal in terms of RNA, or is it sort of one of these, depends on what problems you want to answer? I'm interested in your perspective on that.
And single cell versus bulk, and it's fascinating.
Oh yeah! Oh no! You make up a big
it's broad.
About. I mean, first of all, transcriptomics. You have to be careful about when we do metabolomics and proteomics. We look at what circulates in the blood. When you do transcriptomics in the blood, you look at the transcriptome of the white blood cells. So that already doesn't link to each other. I think the other part where transcriptomics comes in, and a lot of that is GTEx, where you have basic transcriptomics of the organs. And that data is hugely important. And it always gets overlaid with GWAS data and things like that. Whether I'm very much a big fan of transcriptomics in white blood cells, I think there are not many people really doing that. I mean, there have been a few studies. The data is out there. It's in GTEx. It's used. But further than that, I mean, it would be really like, and it's very much confounded with the white blood cells and things like that. We've done work with methylation. So methylation is a good proxy for transcriptomics in a certain way, but it's also in the white blood cells if you don't have access to tissue. And then Cindy mentioned single cell. Now, single cell is a totally different beast, right? I mean, single cell is not something you do on the population level in Cora or Framingham.
Mechanistic biology.
Single cell, I think single cell is basically once you get ideas from GWAS on potential pathways, things like that, then it's the next level where you try to really pinpoint what's going on in these cells and things, things like that. So in the end, I think there's like terms that really need to be put into context. And the same is true for metabolomics and proteomics. Of course, there is metabolomics of cell culture. There's proteomics of tissue extract. And we haven't even mentioned the microbiome. Exactly.
Right? But also, what do you think about, I mean, we've just got a couple of papers that I can think of that have done this, but running, say, proteomics or metabolomics at the same time that you're taking these single cell measurements, these single cell transcriptomics measurements, or site-seq, which also includes the proteomics of the cell to be able to identify cell type, and being able to see over time what's showing up in the plasma. That seems really compelling to me, just in terms of translating something mechanistic into something you might be able to measure in the plasma, but it's all discovery, right?
Well, I think probably the first nearest thing would be single-cell proteomics. I don't know where exactly that stands, but I think doing bulk proteomics and combine it with single-cell RNA and you have it, that is not really something I would favor. There are other techniques where you can have you can have this kind of have like antibodies for this protein surface and have single-cell resolution like that. There's data like that. That, of course, makes sense, but that's also in terminology, is that really proteomics? I mean, you're doing antibodies or surface proteins. So even the term proteomics, you have to be careful.
Yeah, it's single-cell
transcriptomics
where you're able to identify the cell type. That's how I think of it. Although people do call it proteogenomics, but
No. There's also protein things. You can do
from the cell
antibodies.
surface.
And not only the transcript on the cells.
Exactly.
Yeah. That's actually
so then you can determine if it's CD4 positive, CD4 negative. What's the nature of those cells, right? And then I think being able to say in the publication that comes to mind is the Italiano paper on LIF, leukemia inhibitory factor, that where they looked at PD1. I believe it was PD1. An immuno-oncology treatment in responders and non-responders, and simultaneously did proteomics in the plasma. And that, I think, was what I had in mind. And there's maybe 3 publications I know of that had that. And I don't know, I guess I'm biased by trying to figure out what we can measure without biopsying, and being able to make as much use of those precious biopsy samples as we can to then translate it. But you also talked about methylation, and I think I diverted you a bit. I just...
No. I just brought up methylation because we also did not only GWAS, but also epigenome-wide association with the methylation. And there are also signals coming up where I think we in my view, we have this kind of thing like, say, genetic variant. In your metabolome is like, we like to say these are like different valve settings. You have your organism is like all these pipes, and your metabolites flow through the system, and then these valves make you individual. That's your metabolic individuality. And I mean, you don't tune them, you inherit them from your parents.
I guess your and then metabolic your metabolic status, which is based on your genetics and to some extent your environment, is tuning them, I guess was what I was thinking. But
there are 2 things. The one is that comes from your parents, that's your genes. So that's what I say, this valve setting. That's set, and you can change that. And then, of course, the system buffers itself and regulates itself, and then you have other regulators. That could be the methylation, for instance. Total because then there's a certain protein, you need it more or less, and then the methylation adjusts and says, I need more or less of this or that enzyme. And that's something we've seen in some of the studies. And then you have so the methylation, to my view, is a bit the readout of the stress and how your body reacts to disease. And then in that sense, it's very complementary to the genetics. And you can basically get the nature versus the nurture in one measure. And you can do that in population studies with the arrays for genetics and for methylation in the same time, and then combine that. And we've done that. We have papers with Metabolon and with metabolomics and with proteomics on them. And especially for diabetes, all these complex disorders, they're really a nice signal coming out. And parallel, you have positive controls, like smoking is one of the strongest methylation signal. And you see like a handful of genes that are all associated with smoking, and these genes make sense. They actually are in the pathways for detoxification and things like that. So I think there's a lot of promise of combining these things as well, like the methylome and the proteome. And you mentioned also the glycans. So we have glycan data as well, so you can add protein glycosylation on that and find out what's for instance, IgG glycosylation, what's the inflammation state, things like that. Well, and I mean, that's what fascinates me, to get more and more data, and ideally all in the same sample, because then I can correlate the one to the other and really understand how they interact, how they work together. And then the GWAS on all of them, do GWAS on them, because the GWAS in a way is also a way of actually questioning your data. How well is your phenotype? Because a genetic variant can't lie. It's always causal. Other factors can be confounding or whatever, but genetics, except for some very constructed cases, it's always the causal variant. It's
unidirectional, right? You don't have to test for reverse. Very good point.
Yeah, your genes don't change because your metabolism changes.
And I admit, I did open up a big box with that particular question. I want to thank you, Dr. Suhre, for your generous time. I have one final question for you. And how's the weather in Qatar today? It's September. Is it still hot?
Yeah, it's something around 40 degrees, but it will go down, and there will be the Football World Cup coming in November, and then it will be nice, like 25, things like that.
Can I count on you for a after World Cup report in terms of what it was like?
I bet that was
I don't know anybody else. I don't know anybody else in Qatar at the moment, so you will be my Qatar representative.
No, I mean, we are expecting between 1 and 2 million people coming to Qatar. And there are lots of, and you can buy tickets at the moment. It would be very interesting.
Very interesting.
Do you have tickets yet?
No, I'm honestly, I'm not a football fan.
We got to edit that out.
I hate to say that. I hoped you wouldn't ask.
No, it's okay. It's all good. But
Qatar has been preparing for 10 years for this, and I think they really put a lot of. They're really passionate about football.
Yeah.
It's not like in the West very often it's like, oh, Qatar bought the football club, and there's a lot of negative things. Sometimes a bit frustrating to hear my compatriots in Germany, oh, you have to boycott these guys, they're exploiting. It's absolutely, when you're here,
not
it's absolutely
true.
not,
It's
not
passion,
like that.
it's true love.
And especially I think the Emir, he really brought the football cup here because he's a football fan.
Remarkable. And it's
else.
kind and generous of you to leave that ticket for someone who's truly, very keen.
Thank you for the conversation. Right. Really enjoyed it.
So much fun. Thanks, Karsten.
All right.
Bye-bye.
Bye-bye.
Thank you for listening to the Proteomics in Proximity podcast, brought to you by Olink Proteomics. To contact the hosts or for further information, simply email info@olink.com.